Good afternoon. I am delighted to welcome you all to today's webinar. My name is Letícia Mota. I am a process development scientist at APC. Today, we will embark on a journey through the world of chromatography and explore strategies to overcome one of the most common challenges we face in biopharmaceutical manufacturing. In this webinar, we will discuss some principles of downstream processing with a focus on chromatography and maintenance of the biotherapeutic life cycle. Then I will showcase the strategies that we applied at APC to develop a second generation downstream processing to improve process efficiency and productivity. Our goal today is to give you insights and practical solutions that can be applied to your own work. So I encourage you to ask questions in the end. So let's start. The global biotherapeutics market was estimated at around $260 billion in 2021, and it is expected to be worth around $600 billion by 2029. That growth is a consequence of factors like the increase in the prevalence of chronic diseases, higher investments for healthcare infrastructure, and research and development activities. As the demand for biologics continues to rise, so does the pressure on the biotherapeutics life cycle, which includes bioanalysis, biotech transfer, bioengineering, upstream, and downstream processing. Downstream processing is the focus of this webinar, more specifically chromatography. The goal of a downstream process is to remove impurities so that the final product meets the critical quality attributes specifications. Those impurities can be derived from process, for example, cell culture components, HcDNA, HCP, BSA, or they can be derived from the product, for example, aggregates, fragments, and subunits. In order to remove those impurities, a downstream process combines various unit operations and typically requires more than one chromatography step. The first one often is a capture step, in which the target protein binds to the resin while the other components are washed out. The most common resins used in this step are affinity and ion exchange. The role of the following chromatography steps, the intermediate and the polish steps, is to further remove the impurities and increase the purity of the target protein so that it is safe for the patient. After a downstream process is established, changes may happen. For example, materials used in the process become obsolete. We may face some supply chain issues, or maybe we want to incorporate new technologies, or optimize the process, or simply have a backup plan. When that happens, we need to develop a second generation process to maintain the biotherapeutics life cycle and guarantee that patients will keep having access to that medicine. So that's what we did at APC, and I'm going to showcase it now. We have been asked by a client to develop the second generation of a downstream processing for a very complex biomolecule, and we achieved that goal in only 6 months. But to achieve that, first we had to develop a very efficient analytical workflow to support the downstream work. First, we developed a high-throughput reversed phase HPLC method to assess the purity and titer of eluate samples from the resin screening. The eluates were collected into a 96 well plate, and the samples were directly injected for HPLC analysis, with no need for a sample preparation. The HPLC method passed all performance criteria. The same method was able to analyze purity and titer in only 6 minutes. More than 200 samples could be analyzed overnight with that method. This analytical strategy was essential to support the development of the downstream process and to identify the final process conditions so quickly. Across this project, more than 3,000 samples were analyzed. Once the analytical methods were established, the development of the downstream process is started, going from the optimization of the capture step, which happened using the high-throughput screening in 96 well plates and in columns, followed by optimization of the intermediate step, and finally the polish step. All that was completed in only 6 months. In order to achieve a successful chromatography purification, we must evaluate and define some key variables for the process, including mobile phase that can change according to pH, ionic strength, and composition, resins that may vary according to the chemistry, morphology, particle size, and accessible surface area, and the biomolecule. It could be a monoclonal antibody, glycoprotein, mRNA, virus, for example. Regarding the mobile phase, we can have hundreds of possibilities, different pHs, conductivities, composition. So how to define what is the right mobile phase? Well, first, we need to keep in mind that the goal of the chromatography is to separate the impurities from the protein of interest. So it's important to know some protein properties, such as the pi, or isoelectric point. When we have a protein which the pi is different from the pi of some impurity, we can separate them based on their different solubilities. So let's say that the target protein has a pi of 8 and the impurity has a pi of 5. In a buffer with a pH lower than 8, for example pH 6, the protein will be positively charged. So we can use a cation exchange resin to bind that protein. In the same buffer of pH 6, that impurity will be negatively charged, so it will not bind to the CX resin, and it can be removed during the flow-through. Based on these principles, we can predefine the resin type and the pH range of the buffers. Another variable in the downstream process is the resins. It can have different chemistry, different morphology, different particle size, accessible surface area. While selecting the resins that we'll use in a chromatography process, it is crucial to assess the resin scalability, the global availability of that resin, the binding capacity, breakthrough. We need to do some literature research on the resin application for other proteins, analyze the back pressure and flow rate that can be applied to the resin, and verify the robustness. Finally, we need to consider the characteristics of the biomolecule in order to develop the most suitable chromatography process. In the case of monoclonal antibody, for example, the most common resin used in the capture step is a protein A, where the sample is applied onto the column, the protein binds to the resin, the undesired proteins are removed during the flow-through and during the wash step, and finally the target protein is recovered in the elution step. We optimized each of those steps, sample application, wash and elution step, to develop the second generation downstream process. During the sample application phase, we performed a high throughput resin screening in 96 well plates to test the static binding capacity and breakthrough. Then we moved to high throughput resin screening using 0.2 ml columns to test the dynamic binding capacities and breakthrough. The screening of the best conditions for the wash elution step was also done on a 0.2 ml column. Finally, once the best binding conditions had been identified using the high throughput screening, we assessed the dynamic binding capacity of the resins using 1 ml columns in the ACTA. To illustrate our process development strategy, we pre-selected 5 resins according to the criteria explained previously, and we started screening the binding conditions. The resins assessed here were in a 96 well plate format, which allowed us to screen 633 wells, assessing different conditions of load concentration, loading factor, buffer systems, buffer pH, and ionic strength. Once we had identified the resins, buffer systems, pH, and ionic strength that performed better on the 96 well plate, we moved to 1 ml resins to assess the dynamic binding capacity of the resins that performed well previously, and also some new resins that were not available in the 96 well plate format. In total, we screened 5 resins, tested different loading factors, load pH, and conductivity. For that, we used the ACTA Avant 25. That way, we selected one resin only that had the best performance in terms of binding capacity. We also identified the best binding conditions in terms of load density and buffer. So next, we moved to the optimization of the wash and elution conditions. Again, we used the high throughput screening robot, but this time we used 0.2 ml robocolumns instead of 96 well plate. Here, we tested 9 wash conditions and screened 30 elution conditions. Once the best bind, wash, and elution conditions were identified using the high throughput screening, we fine-tuned the method using 1 ml columns in the ACTA system. We started using ACTA Avant 25, then we moved to ACTA 150. That was applied later for the scale-up and proof of concept, using larger columns with representative bed heights. Using this chromatography development strategy, we were able to reduce in 40% the number of chromatography steps, saving process time and costs on resins and increasing the yield. We also identified binding conditions that allowed around 12 times higher load density in one of the chromatography steps. Compared to the first generation process, the new process increased the yield significantly. And very important, the process was developed in only 6 months. The process efficiency increased drastically compared to the first generation. The maximum number of batches per year has increased by 150%. The process time has been reduced by 15 days. The quality control delays decreased from 18 to only 2 days. And the new process can efficiently remove the undesired impurities, achieving the critical quality attribute targets. In terms of process capacity per year, the new process is able to increase the production output by 275%, resulting in a substantial revenue increase. At the same time, the new process reduced significantly the footprint, the labor needs, and cost of goods. In conclusion, in today's webinar, we navigated through the strategies that we have used to develop a very efficient second-generation downstream process. We discussed about some chromatography principles, which are essential to develop a robust process. Then we talked about the high-throughput analytical and resin screening methods that we used, which were crucial to assess as many resins and process conditions as possible in a short period of time, saving us time and resources. The result is that not only the new process added a huge value to the client in terms of yield, time, and cost, but also it accelerated the delivery of life-saving biologics to those in need. With that, I would like to thank you for joining us on this journey to a more efficient downstream process, and I am happy to answer any question.